Very delighted to welcome Ashley Watson here from the group that is working closely with Huri Maharena, who we know well. We brought him into the fold of our Alzheimer's Center, and we'd love to bring Ashley in as well. So this is a great introduction to our folks. How you doing? Good to see you. A few folks coming over. Ashley started with her bachelor's and her PhD at the University of Western Ontario, but then moved to MIT to Lee Weight Size Lab, where she met Hurui. And she was working on first a lot of DNA work and manipulation using CRISPR, and was looking to investigate tau in Lee Weight Size Lab. Then she went off to Stem Cell Inc. or something, right? What's the name? Stem Cell Inc. Stem cell technologies for a little bit in the industry, but that actually was quite helpful in further advancing her CRISPR expertise. And thankfully, Hurui was able to connect with her scientifically to bring her back to San Diego, where she is also applying some of this work within the iPSC models. So let's hear about it. And thanks and welcome.
Awesome. Okay, well, thank you, Jim, for the intro. And please let me know online if you can't hear me properly. I'm really thrilled to tell you today about our work. So as Jim mentioned, in my career, I've studied both neurodevelopment and neurodegeneration. And in that, I've really been struck by the fact that the same mechanisms that act to build the brain often go awry and become dysfunctional in neurodegeneration. And I think the story I'll tell you about today is a perfect example of this phenomenon. And so in the first part of my talk, I'll tell you about our unexpected finding that a senescent-like state contributes to brain barrier development. And then in the second half, I'll tell you why I think this discovery is very relevant to the study of aging and neurodegeneration, and how we're trying to use this knowledge to gain insights so that we can build better therapeutic tools to improve brain health across the lifespan. So I know I don't have to convince you all that neurodegeneration is a huge problem. Our population is aging, and the single greatest risk factor, for neurodegeneration is aging. And over the age of 65, about 1 in 10 individuals will be diagnosed with dementia, and this number is expected to increase because of our aging population. But neurodegeneration isn't simply an inevitable consequence of aging, but rather a loss of homeostatic mechanisms that contribute to brain function and health across the lifespan, and a buildup of cellular and molecular damage. And so some of these homeostatic mechanisms that help to keep our brain healthy include inflammation. And this is a double-edged sword. So acute inflammation can be really protective for brain function, whereas chronic neuroinflammation that is often mediated by the immune cells of the brain, microglia, can be detrimental to brain health. And this is strongly linked to neurodegeneration, wherein microglia move from a state of homeostatic surveillance towards an activated pro-inflammatory state that is detrimental to brain function. Moreover, our brain is a highly vascularized organ, and the vasculature system of the brain helps to provide nutrients and oxygen to different parts of the brain for brain function. And at the interface of the bloodstream and the central nervous system, it's something known as the blood-brain barrier. And so this is a highly selective interface that allows molecules from the periphery into the brain, and as I mentioned, helps to transport oxygen and nutrients into the brain. And it's comprised of endothelial cells that form blood vessels, and then parasites that wrap around endothelial cells to provide stability for the blood-brain barrier, as well as astrocytic end feet that provide metabolic coupling to this barrier. And decades of research now has shown that this barrier breaks down with age, and in cases of neurodegeneration, potentially resulting in a leaky barrier and impaired brain health. Another barrier within our brain that's highly relevant is the blood cerebral spinal fluid barrier, or the blood CSF barrier. And the choroid plexus is a structure that is at the interface of this barrier. And so the choroid plexus is found in each one of our brain ventricles, and it produces cerebral spinal fluid. It's comprised of choroid plexus and epithelial cells that line the outside of the choroid plexus that are multiciliated cells that I mentioned produce CSF. And there's a lot of interplay between these cells and the vasculature of the choroid plexus in this CSF production. And this system is implicated in brain clearance mechanisms. And so over time and during the day, there's a buildup of metabolic waste within the brain, including toxic proteins like amyloid and tau that then become cleared, and the CSF bathing of the brain is part of this process. And so it's highly relevant to neurodegeneration. And then finally, something that links all of these systems together is sleep. And so the permeability of both the blood-brain barrier and the blood-CSF barrier changes dynamically throughout the day and is influenced by sleep-wake cycles. And CSF production itself is also influenced by sleep-wake cycles. And sleep is both, or I guess disrupted sleep is both a cause and a consequence of neurodegeneration. And so I'm really interested in how all of these systems work together to promote risk for this devastating condition. So while these things are all quite well established in neurodegeneration, another emerging mechanism is cellular senescence. And I'll tell you in a few slides exactly what senescence is, but numerous studies now have shown that senescent cells accumulate with age in a number of different organs, including the brain, and also accumulate in response to pathological cues such as amyloid and tau. And in particular, the cell types that exhibit this phenotype are glial cells and brain endothelial cells that show features of senescence in most models of neurodegeneration and in humans. And so this data is from the P30 tauopathy model, where they showed that brain endothelial cells exhibit markers of senescence, as well as something known as the senescence-associated secretory phenotype, which I'll discuss in a couple slides. And then this is from humans with AD showing an increase of the senescence marker P21 in microglia in the brain. And why I think this is so fascinating is because a study showed that genetic removal or pharmacological removal of senescent cells can improve neurodegeneration in the P30 model, which shows neuron degeneration in the dentate gyrus of the hippocampus, which is restored after ablation of senescent cells back to something that looks like control cellularity. And then also cognitive performance in this model was rescued upon ablation of senescent cells, suggesting that senescent cells might be a really attractive therapeutic target for neurodegeneration and the reduced cognitive performance in these disorders. So what is cellular senescence? So here we have a happy little cell, and when it gets exposed to any sort of stress, it will enter, or such as telomere erosion, DNA damage, oxidative stress, irradiation, or even prolonged replication, it will enter a state of permanent cell cycle arrest known as senescence. And this state is characterized by upregulation of a number of cyclin-dependent kinase inhibitors, including P16, P21, P53, which act to reinforce this growth arrest. And then also is characterized by a number of other features, including upregulation of anti-apoptotic machinery to allow these cells, even though they're not dividing, and they upregulate a lot of these key proteins that are involved in the decision between cell cycle arrest and cell death. These cells do not undergo apoptosis because of the upregulation of anti-apoptotic machinery. And then the cells also exhibit profound metabolic changes, including changes to autophagy and lysosomal and mitochondrial expansion. And in fact, the lysosomal expansion in senescent cells is one of the most well-known markers of senescence. Senescence-associated beta-gal takes advantage of this lysosomal expansion and can be used as a marker for senescence. Another key feature of the senescent state is that the cells, regardless of what kind of cell it is, will acquire a pro-inflammatory signature. And so the cells upregulate pro-inflammatory pathways like NF-kappa B, and this transcription factor will upregulate genes that encode secreted factors that are part of the senescence-associated secretory phenotype, or the SASP. And these secreted factors include growth factors and pro-inflammatory cytokines and matrix metalloproteases that act to both reinforce senescence through autocrine signaling and can also change the cellular environment through paracrine signaling. And so I've already told you that aging and neurodegenerative cues like amyloid and tau can induce this state, but we were really curious about a finding that was published over 10 years ago that developmental cues can also induce this state. And so these two papers published in 2013, back-to-back in Cell, showed using senescence-associated beta-gal staining on whole mount embryos during mid-gestation that there was really distinct, staining of the senescence marker. And so they saw SA beta-gal kind of lining the spinal cord of embryos within the optic vesicle, which is part of the inner ear, the developing kidney, as well as the tip of the tail, the neural tube, and they found senescent cells also lining the tips of the developing limb. And these papers characterized developmental senescence and found that it's actually P21 dependent and not dependent on other cyclin-dependent kinase inhibitors that are involved or implicated in senescence. And they showed this by demonstrating that senescent or embryonic senescent cells that stain positive for beta-gal also stain positive for P21. And when you genetically remove P21, you get a reduction in this senescence. And furthermore, they showed that in the context of P21 knockout, that development of the limb and development of the kidney was impaired, suggesting that senescence plays a role in embryonic growth of tissues and patterning of these tissues. And so we were curious whether embryonic senescence plays a role in brain patterning and morphogenesis, because they showed that you can actually detect senescent cells in the developing neural tube, but they didn't really go any further than that. So we wanted to characterize this further. And so in order to do this, we performed P21 immunostaining after sectioning through an entire E13.5 embryo. And the reason we chose E13.5 was because in those initial studies, they showed that embryonic senescence is transient and it seems to peak during mid-gestation around E13.5. And so we sectioned through an entire embryo, stained it for P21, and then annotated all the brain regions where we saw P21 positive cells. And this is what an entire embryo stained for P21 looks like. And as you can see, there's a really distinct staining pattern that kind of mirrors what they saw by SA beta-gal, where you get P21 staining along the spinal cord in a variety of organs, including the liver and the heart. But when we look closely at the brain, we saw that P21 positive cells seem to, or there seemed to be a higher degree of P21 positive cells in really particular brain regions. And these corresponded to the choroid plexus of the lateral ventricle here and the fourth ventricle. When we did coronal sections, we also saw P21 staining in the third ventricle choroid plexus. And then we also saw that there was a high concentration of P21 positive cells in the cortical hem and the prethalamic eminence, which is right next to the lateral ventricle choroid plexus. So this is pretty fascinating because I guess as a developmental neurobiologist, at least during my PhD, that's what I was trained in, I kind of expected to see low levels of P21 everywhere, especially in the developing cortex, because at E13.5, this is when cortical neurogenesis is going on. And previous studies had kind of implicated P21 in cell cycle exit of these neurons. And so I was really surprised by what we saw. And a very talented research associate, Zoe, who's now doing her MD PhD at UCLA, performed a thorough characterization of the P21 positive cells throughout the embryonic period. And she found that in the cortical hem, there was a distinct peak of P21 positive cells, and they disappeared by E18.5 right before birth. Whereas in the choroid plexus, we see that P21 positive cells occur in both the lateral ventricle and fourth ventricle choroid plexus, kind of as soon as these structures are developing around E11, E12.5, and that P21 positive cells are maintained in these structures all the way through to E18.5. So as I mentioned previously, senescence is quite a complex state and no one marker can define the senescent state. So we also wanted to perform SA beta-gal staining to see whether these P21 positive cells also stain positive for another marker of senescence. And when we performed this staining, we saw that curiously, the P21 positive cells within the cortical hem did not stain positive for SA beta-gal, but the cells within the choroid plexus did. And as soon as the choroid plexus formed, there was really strong SA beta-gal staining within this region that corresponded to the appearance of P21 positive cells. So this is pretty fascinating. We were seeing a couple different markers of embryonic or of senescence in these embryonic cells, in these embryonic structures, but we wanted to get a more comprehensive idea of what this cell state is and whether it has any role or any function in brain development. So to do this, a graduate student in the lab, Grace, took advantage of a previously published single-cell RNA-seq data set from Sten Lenarsen's group, published in 2021, where they took mouse embryos from E8 all the way through to E18.5, dissected out the forebrain, midbrain, and hindbrain, and sequenced a ton of cells. And so this is a really rich data set of almost 300,000 embryonic brain cells. And so Grace clustered these cells and recapitulated what the previously published data set showed, which is that there are about 50 clusters of transcriptionally distinct cell types within the developing brain, the majority of which are neuronal origin, and that's these different clusters here. And then we were also able to see some blood cells, vascular cells, and macrophages. And so we then overlaid P21 expression on this map to see if there were particular cell types that expressed high levels of P21. And when we did this, what we saw was actually that, not a lot of cells express high levels of P21, and that there's really distinct places within this UMAP that express relatively high levels of P21. And these clusters corresponded to the choroid plexus, which was awesome. That's what we had seen by immunostaining, and we were seeing it also at the transcriptional level. It corresponded to Cajal-Retzius cells, which I'll get back to in a second, as well as a proportion of CNS-resident macrophages, including both microglia and border-associated macrophages, had a relatively high population of cells that expressed a high degree of P21, and vascular cells as well that included pericytes and endothelial cells. And so getting back to the Cajal-Retzius cells, so this was really fascinating because the region that I told you about before, the cortical hem and prethalamic eminence, is the birthplace of Cajal-Retzius cells. And so we did a bit more of characterization of these P21 positive cells and saw that indeed they seem to co-express some markers of Cajal-Retzius cells. So Cajal-Retzius cells are a very specialized neuronal subtype. They're born early in development, one of the earliest born cortical neuronal subtypes, and they migrate from the cortical hem or from the prethalamic eminence through the marginal zone of like the first layer of the cortex and help with cortical neuronal migration. And so what we were seeing was that P21 seemed to be particularly expressed in the cells right before they begin to migrate and upregulate mature markers of Cajal-Retzius neurons, TBR1 and relin. And so potentially indicating that P21 expression here is linked to differentiation of these cells. In the choroid plexus, we saw that the P21 expressing cells were lining the outside of the choroid plexus and stained positive for the choroid plexus epithelial marker, aquaporin-1. And then we were able to detect P21 positive microglia within the brain parenchyma, as well as P21 positive CD206 positive border-associated macrophages throughout development. And then the vascular cluster, as I mentioned, contained both CSPG4 positive pericytes, as well as CD31 positive P21 positive cells. And often the P21 positive cells in the developing vasculature look to be of this, endothelial cell state known as the tip cell state, which is a state where the cells begin to elongate their filopodia and migrate towards angiogenic cues. And it's really important in angiogenesis, or these cells are really important in angiogenesis in the brain. So this is pretty fascinating. We were able to, or I didn't really mention, but earlier when we were performing the P21 immunostaining, we were seeing some random P21 positive cells outside of those three main regions that I described. And now we were able to tell that those cells are likely either macrophages or vascular cells. And so the next question we had was, does this transcriptional signature of P21 high cells from each of these different identities resemble senescent cells? So to address this, we took another previously published data set from mouse embryonic fibroblasts from these two papers, where they induced senescence using a variety of different stressors, including doxorubicin, ionizing radiation, oncogene-induced senescence, and replicative senescence. And the reason we wanted to take all of these different types of senescence is it's known that the state of senescence depends, or I guess senescence is kind of a elusive state. There's many different qualities depending on the stress that induces it. And so we wanted to be really comprehensive in this analysis. At the same time, we took our 4 different cell clusters and we isolated out cells that express a high level of P21 and cells that don't express any P21. And then we performed pseudo-bulk differential gene expression analysis to identify the transcriptional signature of the P21 high cell state by identifying genes that are up or down-regulated in the P21 high cells. So then with all of those gene lists, we performed a randomized permutation test to ask whether there was a statistically significant overlap between any of those lists and any of the genes that are up or down regulated in senescence. And so this is the results of that. So this is a bubble plot wherein the size of the bubble indicates the degree of overlap and the color of the bubble indicates the significance. And so black indicates a highly statistically significant p-value. And hopefully what you can appreciate is that in P21 high choroid plexus epithelial cells, vascular cells, and macrophages, we saw a really strong correlation with the transcriptional signature of senescent mouse cells. And it was directional. So genes that typically go down in senescence were going down in the P21 high state of these 3 different cell types, and the same thing with upregulated genes. But this was distinct from what we saw in Cajal-Retzius cells. So when we compared P21 high to P21 negative Cajal-Retzius cells, the genes that typically go up seem to be correlated with the genes that go down during senescence, overall suggesting that P21 high choroid plexus epithelial cells, vascular cells, and macrophages exhibit a senescent-like transcriptional signature, whereas Cajal-Retzius cells do not. And I kind of mentioned to you previously, but we wanted to know how were the P21 high choroid plexus epithelial cells, macrophages, and vascular cells similar to senescence? So to accomplish this, we used the single-cell RNA data set and performed a gene ontology analysis. So just with senescent cells, what we see is their transcriptional signature reflects the, qualities that we know that are really well established in senescent cells, including downregulation of replication and protein translation, and upregulation of a variety of things, including genes related to the SASP, like secreted proteins, pro-inflammatory cytokines, and the NF-kappa B pathway, as well as upregulation of a variety of other pathways related to fluid transport, including a lot of ion channels, cell-cell interactions, including extracellular matrix genes and cell adhesion, tight junction genes. And then, strikingly, these cells are just mouse fibroblasts induced into a senescent state in a dish, and they upregulate a lot of genes related to vasculature development and things related to angiogenesis. Okay, so then we wanted to see in the P21 high state of those 3 other cell types, how did they resemble senescence? And so this is what it looked like. So it looks like those 3 different cell types resemble senescence, sometimes in similar ways and sometimes in quite unique ways. The similarity occurs with the downregulated gene set, wherein all 3 of these cell types in their P21 high state seem to suppress cell cycle, DNA replication, and protein translation, whereas the upregulated genes were quite distinct between choroid plexus epithelial cells in the P21 high state and then vascular and macrophage cells. So I'll get into those differences. So in choroid plexus epithelial cells, the P21 high state was characterized by upregulation of genes related to fluid transport, such as ciliogenesis genes, things related to fluid movement, as well as extracellular transport and ion channels, as well as upregulation of tight junction genes and things related to circulatory system development. So I think this is telling us something about the function of this P21 high state. And so if we go back to the choroid plexus, a structure that is primarily comprised of these epithelial cells that line the outside of the choroid plexus and are bound together by tight junctions to create this barrier. They also interact a lot with the vasculature during development to vascularize the structure and also to provide the raw materials for CSF. The cells are highly ciliated, and this helps increase surface area and enhance CSF production. And so the fact that we were seeing upregulation of ciliogenesis pathway, as well as tight junction genes, was, I think, indicating that this P21 high state is associated with development of the structure, as well as its function potentially in CSF production. Also, we saw upregulation of circulatory system genes, and many of those encode secreted factors that actually are well-known angiogenic cues. And so potentially, I think this P21 high state may be signaling to the developing vasculature to vascularize the choroid plexus structure. And this includes VEGF, adrenomodulin, DLL3, and JAG2. And then in the P21 high vascular cells and macrophages, we saw something quite distinct where the fluid transport upregulation was not occurring in these cells, but they did seem to exhibit a strong SASP-like signature, where they had enrichment of secreted genes in their upregulated gene list, as well as NF-kappa B pathway genes. And then they also upregulate things related to cell-cell matrix interactions and vasculature development. When we took a closer look at the SASP of these cells, what we saw was many classical SASP factors were upregulated in the P21 high vascular cells and macrophages, and this was not observed in the choroid plexus epithelial cells. But then we wanted to do a more unbiased analysis of this. And so we took all of the secreted protein or secreted genes that are upregulated in senescence and asked whether there's enrichment of those factors in the secreted genes that are upregulated in these two different cell types. And we found that the secreted genes that are upregulated in either P21 high vascular cells or macrophages, there's a strong degree of overlap with those secreted factors upregulated in senescence, suggesting that this is potentially a SASP within these cells. And then before I get into what I think the function of the P21 high state is, I just want to tell you a little bit more about brain vasculature development. And so the majority of the brain vasculature develops from sprouting angiogenesis. And I mentioned previously these tip cells that have filopodia that migrate towards angiogenic cues. And then following behind these cells are stalk cells that proliferate and help to elongate the vessel. And there's a lot of crosstalk between these two cell states in the endothelial population, but also at the same time as the vasculature is developing, the endothelial cells send out signals to recruit other cell types like parasites and macrophages to help with stability of the newly formed vessels, as well as remodeling of these newly formed vessels. And interestingly, what we were seeing was the SASP of the P21 high vascular cells seem to be encoding many factors that are involved in recruitment of both parasites and macrophages. And these included some of the most well-known secreted molecules that are involved in this process. So in the P21 high endothelial cell state, we were seeing upregulation of things like PDGF-beta, which is a known cue for parasites to help with their survival and attraction towards developing blood vessels. And some other factors. And then we were also seeing secretion or upregulation of genes encoding secreted factors that help to attract macrophages or microglia towards developing vessels, including CSF1, IL-34, and TGF-beta. Furthermore, we were seeing that we performed, analysis to try and identify the receptors that these ligands bind to. And we saw that, for instance, in macrophages, there was an enrichment of the receptors that the upregulated SASP factors bind to, suggesting that there might be bidirectional or communication between this P21 high endothelial state and macrophages in the developing brain. Furthermore, when we looked at the SASP of the P21 high macrophages, we saw a number of secreted factors that encode well-known pro-inflammatory molecules like IL-1 beta, IL-6, TNF alpha, as well as things related to remodeling of the developing vasculature like PLAU and IGF-1. And we saw upregulation of the receptors for these secreted factors in the P21 high vascular cells, together suggesting that there may be some bidirectional communication between these P21 high cell states in these two different types of populations, potentially in mediating vasculature development. And so far, what I've shown you is that the transcriptional signature, at least, of the P21 high state suggests that P21 high choroid plexus epithelial cells may participate in blood CSF barrier vascularization, integrity through upregulation of tight junctions, as well as the function in producing CSF. Whereas in the vasculature or vascular and macrophage P21 high state seems to potentially be involved in facilitating interactions between macrophages, endothelial cells, and parasites to enhance vascular integrity and maturation, and potentially the pro-inflammatory state of the P21 high macrophages contributes to endothelial cell migration and vessel remodeling. So this is pretty cool, but so far it's all correlative. And one thing we wanted to know was, when do these P21 cells appear during development? And we got some clues from the single cell data set, but we also wanted to do some lineage tracing to see when do they appear and do they maintain and persist into the brain or into the postnatal brain. And so we generated a mouse model wherein estrogen responsive Cre is downstream of the P21 promoter. And we did this in collaboration with the WashU Genome Editing Core and the UCSD Transgenic Core Shared Resource. And then we crossed this strain with a inducible reporter strain, wherein a stop cassette is flanked by LOXP sites, such that when active Cre is present, you'll get expression of a reporter, and our reporter is TD tomato. And so we performed windows of tamoxifen injection in the early embryonic period from E9 to 11, in the mid-gestation period at E12 and E13, and in the late period at E14 and E15, to see whether there was distinct spatiotemporal dynamics of P21 expressing cells during brain development. And then we analyzed the embryos at E18.5 right before birth, and then at postnatal day 21 to ask whether these cells persist into the postnatal brain. And so the next slide has a lot of data, but I'll walk you through it. Basically, we were able to recapitulate what we saw by single cell data analysis, that these, and by immunostaining, that P21 expressing cells seem to appear primarily during mid-gestation and seems to be regional, where in the choroid plexus, the cells appear at around E12, E13 and increase in frequency over time and persist into the postnatal brain. So we saw a relatively equal frequency of these cells at both E18 and postnatal day 21. So cells or choroid plexus epithelial cells that previously expressed P21 in the embryonic period were still maintained in the adult brain. And we saw something similar with vascular cells and macrophages, but the proportion of P21 positive cells was higher in the subcortical regions like the thalamus and the hindbrain relative to the cortex of these cell types. But these cell types seem to be particularly appearing at around E12, E13, and E14, E15, which is coinciding with when the vasculature is really maturing and the blood-brain barrier is forming. So we concluded from this that P21 expressing cells seem to emerge at critical time points during development and maturation of the blood-brain and blood CSF barriers. And taken together with the transcriptional information, it's kind of putting together a picture that these cells may be participating in establishing these barriers during embryonic development. But again, all correlative. So we really wanted to know, could we get rid of these cells and see disruptions in these barriers? By asking, what is the function of these P21 expressing cells? So to do this, we used our P21 Cre model, and we crossed these mice to inducible diphtheria toxin expressing mice. And so whenever Cre is active, diphtheria toxin will be expressed. And as the name suggests, it's a toxin and will cause cell death. And so in order to control for any leakiness of the diphtheria toxin, we set up mating strategies so that we would have Cre negative controls that had the diphtheria toxin gene but did not express Cre, and then P21 ablated mice that express Cre and diphtheria toxin. And we performed tamoxifen injections during the peak of when we saw these cells between E11 and E15.5, and then looked at the embryos at E18. And a lot of this work was done by Kenzie, who's a research associate in the lab, as well as John, an undergrad in the lab. And so if you remember back to that first picture I showed you of P21 expressing cells throughout the entire mouse embryo, there's lots of cells that express P21, not just cells in the brain. And so I was quite skeptical that this would even result in a viable embryo. But what we saw was that there is viable embryos. They do not survive postnatally, and they have a very distinct phenotype. They have a phenotype of stunted growth relative to controls, as well as a hemorrhage phenotype. So you can see a buildup of blood, especially kind of in the back part of the brain and along the spinal cord of these embryos. And it doesn't happen in every embryo. For instance, this is an embryo that doesn't really have a strong hemorrhage phenotype, but there is a pretty prominent hemorrhage phenotype. When we dissected out the brains, we also saw a hemorrhage phenotype within the brains of the P21 ablated mice relative to controls, and that's just quantified here in terms of hemorrhage severity. So as you can see, it's kind of a spectrum, but sometimes the hemorrhage was really severe. So we took a closer look at this by staining for erythrocytes or red blood cells in sections from P21 ablated E18 brains and controls. And what we found was that we could see a buildup of TER119 positive erythrocytes around the outside of the brain, which kind of matched what we were seeing visually. But then also we could see evidence of cerebral hemorrhage, so erythrocytes or red blood cells outside of blood vessels, as well as a buildup of erythrocytes in kind of more larger blood vessels. And that these two phenotypes kind of appeared in the P21 ablated embryos more so than controls. When we took a closer look at the sites of hemorrhage, or where we saw red blood cells outside of the blood vessels, we saw evidence of rupturing of the blood vessels, as well as reduced staining for the tight junction marker ZO1, which is typically at E18.5, highly expressed in the vasculature and is associated with integrity of the blood vessels. And we were seeing a reduction in the staining in the P21 ablated embryos. So together, this suggests that ablation of P21 expressing cells seems to impair integrity of the blood-brain barrier and result in cerebral hemorrhage. And I think we are really interested in taking a closer look at the P21 high state to see if there's other pathways that we can target just specifically in that state to modulate it, as opposed to just kill these cells entirely. Right, so then another prominent phenotype that we observed in the ablated mouse brains was a reduction in ventricle size, as well that was accompanied by a reduced size or length of the choroid plexus structure. So these are coronal sections, and you can see in the control, we have a pretty large ventricle that's filled with cerebral spinal fluid that is being produced by the choroid plexus. And then in the P21 ablated brains, unsurprisingly, because we know that P20 or the choroid plexus epithelial cells express P21, there's a reduced size of the structure, and the ventricle is also significantly reduced in size. Another thing that we saw was that the P21 ablated embryos had this really interesting swelling phenotype. They kind of looked like a balloon compared to the controls, which was really odd. And I had no idea how to interpret it when I first saw it. But I think it could indicate fluid dysregulation systemically throughout these embryos. But even more so what we found, and then we wanted to also, in a different way than just Nissl staining and quantifying the area of the ventricle, we wanted to use other methods to quantify the volume of the ventricle, which is a surrogate for the amount of CSF that is being produced. And so we collaborated with David Barry at this Center for Functional MRI here at UCSD and performed MR scans or MRI images of the P21 ablated and control embryos. So that's a sagittal MR scan. And then what I'm going to show you on the next slide is some coronal images. And I hope you can appreciate that the size of the lateral ventricle is reduced in the P21 ablated embryos compared to control. And in these T2 weighted images, you can see that fluid is brighter, has a brighter signal than tissue. And so we were able to then quantify the volume of the lateral ventricle and the fourth ventricle, and saw that after ablation of P21 expressing cells, that these volumes were reduced. Moreover, what we saw was we were able to kind of see by MRI that the fluid that was being built up outside of the brain. And that's kind of indicated here and quantified here, just looking at the intensity of the MR signal here and an identical ROI in the control. So globally, this suggests that ablation of P21 expressing likely the choroid plexus epithelial cells is resulting in a reduced volume of the lateral ventricle and presumably also reduced CSF production. Looking closer at the choroid plexus structures, what we saw was kind of a general disorganization of these structures in P21 ablated mice compared to controls. And when we stained with ZO1, that tight junction marker, in controls, you usually see this characteristic cobblestone pattern of the epithelial cells. And this was, ZO1 staining was really mislocalized in the P21 ablated embryos. Moreover, staining with phalloidin, which stains F-actin and can help indicate polarity of these epithelial cells, which is really critical for proper function and production of CSF. We saw that there was reduced phalloidin staining kind of in the interior of these structures where the vasculature typically resides. And we saw that the vasculature was reduced in area and also intensity of the stain of CD31 in the choroid plexus structures of P21 ablated embryos compared to controls. So together, all of this data, I hope, convinces you that the senescent-like state that's characterized by upregulation of P21 is required for development of the blood CSF and blood-brain barriers. And when we remove these cells, we get impaired development and function of both of these barriers. And so I think that this suggests that in the developing brain, senescence is not just a pathological process, but it's rather a carefully orchestrated mechanism that contributes to barrier formation and function. I think this finding kind of challenges the traditional view of senescence as being merely a pathological process. I also think that it kind of raises another critical question, which is in aging and neurodegeneration, could these same pathways become dysregulated? So I mentioned in the beginning that these barriers are dysfunctional in aging and neurodegeneration, and that contributes to risk for this disease, and that also sleep plays into all of these things. So in order to start addressing this question, which is really where I want the work to go in the next phase, we wanted to ask, how transient is this senescent-like state? Does it just occur during development, or does it occur throughout the lifespan? And so we used our reporter system to perform daily tamoxifen injections for 5 days at 3 months old. So when the mice are young adults, but their brains have already developed. And what we found was that we were unable to detect any TD tomato positive brain vascular cells or macrophages, but the choroid plexus still retained some P21 expressing cells. And even more surprisingly, the choroid plexus still expressed SA beta gal. So it's still expressing this additional marker of senescence and looks to be expressing P21. And we were able to see some TD tomato positive cells. When we took an even closer look at the choroid plexus and the TD tomato expressing cells, we saw something super interesting, which is that after this 5-day injection, we can see TD tomato positive cells that still express P21, but then we also saw TD tomato positive cells that no longer express P21. And we saw choroid plexus epithelial cells that expressed P21, but had not expressed P21 in the last 5 days. So this suggested that P21 expression in the choroid plexus is not static, but it actually fluctuates over time. And I thought this was super cool. So getting my brain going to think of what could be regulating P21 expression in this structure. And so this brought me to a wealth of literature that shows that the choroid plexus is actually an important component of the circadian clock. And this study from over 30 years ago that showed that CSF production is regulated through sleep-wake cycles, and it's heightened during the day. And the function of this CSF production changing with sleep-wake cycles is thought to be related to brain clearance mechanisms. So during the day, when our brains are active, our neurons are firing, we're getting a lot of metabolic waste buildup. Our norepinephrine levels are also high, and our blood vessels are dilated, providing more raw materials and enhanced production of CSF. Further, the choroid plexus is more active during the day. This buildup of pressure from the increased CSF production during the day is really essential for what happens at night, which is that we have low norepinephrine, our blood vessels constrict, there's more space between the blood-brain barrier and the cells of the brain, and this helps to flush out metabolic waste, and lymphatic clearance pathways are highest when we're sleeping at night. And so I started to think that potentially the P21 positive state could be regulated by sleep-wake cycles and could be involved in controlling the different functions of the choroid plexus in the night and in the day, especially based on what we were seeing embryonically, where the P21 high state seemed to be associated with upregulation of tight junctions and changes to CSF production. So to address this question, we took again our reporter strain and we performed either a morning injection of tamoxifen at 6 a.m., right before the rest phase of mice, since they're nocturnal. And then we also performed in a separate mouse tamoxifen injection at 6 p.m. right before the active phase. And strikingly, what we saw was that the TD tomato positive cells was distinct between the morning and the night, where we saw many more TD tomato positive cells or P21 expressing cells in the rest phase relative to the active phase. This does suggest that potentially P21 expression is being regulated by sleep-wake cycles or controlled by the circadian rhythm, and it could have some function in CSF production and metabolic washout in the brain. So this is all very preliminary data. And while we don't yet know the molecular characteristics of these P21 expressing cells in the adult choroid plexus, like I said, if we take what we learned from the embryonic P21 high cells, it may be suggesting that this P21 high state is associated with upregulation of tight junctions, reinforcing the barrier, and helping to regulate this washout of metabolic waste and increase CSF production during the day. So we're actively investigating this now. But getting back to the vascular cells and macrophages and microglia, as I mentioned, we do not see that they express P21 in a wild-type adult mouse, but I hypothesize that mechanisms that regulate senescence during development may also become reactivated in neurodegeneration. So to address this hypothesis, we've turned to the P30 tauopathy model, which I told you in the introduction, exhibits neuroinflammation and neurodegeneration. And also studies have shown or have identified senescent microglia and endothelial cells in this model. And furthermore, when you remove senescent cells in this model, you get cognitive improvements. And so potentially the senescent state is enhancing inflammation and enhancing brain degeneration in this model, and might provide a really attractive therapeutic target. So to start looking at this, we performed staining for, so then we stained for IBA1, P21, and beta-gal, and even at 9 months, we see that the choroid plexus still expresses beta-gal and still expresses P21. We were also able to see that in the P30 model that there's more microglia, which is consistent with the literature, knowing that there's inflammation or neuroinflammation in this model. And we were able to see evidence of P21 expressing microglia in the P30 model at 9 months, which is consistent with the literature. And so now we're really interested in knowing whether these cells are the same cells that go through embryonic senescence and whether those cells that go through embryonic senescence are more vulnerable to the senescent state in the context of neurodegenerative cues like hyperphosphorylated tau. When we took a closer look at the choroid plexus, we see that this is very preliminary, so take it with a grain of salt, but there does seem to be differences in expression of P21 in the choroid plexus between the wild type and the P30 model. And so now we're really eager to explore this further, and to look to see, we're crossing this model with our reporter strain so that we can see which cells express P21 in this model. Are they the same cells as we see embryonically? What is their characteristics, and can we modulate that state for any sort of benefit to brain health? So just to conclude, I hope I've convinced you today that a senescent-like state contributes to development of brain barrier systems. And now we're really trying to investigate whether the state also is associated with dysfunction of these same systems in aging and neurodegeneration. And so we're leveraging our mouse model as well. In the lab, we do a lot of human stem cell culture and generating human stem cell derived models of neurodegeneration and different types of organoids like choroid plexus organoids to see if we can see in a human system the same kind of thing. And then the overarching questions that are driving the next phase of this research include whether embryonic senescence influences lifelong neurodegenerative disease risk, if embryonic senescence primes the brain for barrier dysfunction and neuroinflammation in aging, if we can target this system to protect the blood-brain barrier and potentially mitigate neuroinflammation in neurodegenerative disease, and whether we can potentially harness circadian regulation of P21 to optimize CSF production and waste clearance to reduce neurodegenerative pathology. So these are really broad and lofty questions, but that's where I see this work going, and I'm really excited to pursue it. Okay, so just to thank everyone that was involved, I think I shouted out the majority of people that provided a lot of help with this work, like Zoe, Kenzie, Grace, John, as well as the rest of the Maharena Lab. This project was entirely funded by startup funds from the School of Biological Sciences to Harui when we started the lab 3 years ago. And I'd like to thank our collaborators, especially Dave Barry at the CFMRI here at UCSD, and then the Transgenic Core and WashU. And last but not least, I'd like to thank Harui, who has brought me on this exciting journey, studying this super cool project. And it's just so much fun to be able to do science with one of my best friends every day. So yeah, thank you everyone for listening.